there's a overproduction of excitatory synapses in the visual cortex. So they will peak at about 150% of the adult value in early childhood. And then there is a mad process where those synapses are whittled away during adolescence. So you lose about 50% of those synapses during adolescence as you're fine tuning your connection.
So how do we get to the plasticity in the story?
Oh, thank you. I'm so relieved to hear that question. It's been a long and really fun journey for me, my studies of plasticity, but it really begins with, again, Hubel Wiesel, David Hubel and Torsten Wiesel. And they, at least the way I tell the story, I think this is more or less accurate, is in pondering the precision required to have the precise mapping of the inputs from the two eyes onto common target neurons in the cortex. That is to say, let me just go back to the receptive field concept for a second. So when I talk about a ganglion cell receptive field, it's going to, that ganglion cell will respond to stimulation only of that eye, right? Right. If it's a right eye ganglion cell, you're not going to get a response by stimulating the left eye. You're only going to get a response by stimulating the right eye. When you go to the lateral geniculate nucleus, same story. You've got neurons that have receptive fields. And in fact, those receptive fields do resemble retinal ganglion cell receptive fields. But again, typically those cells will respond only to stimulation of one eye or the other eye, never to both. When you get to the cortex, then the miracle occurs that you find individual neurons that respond to stimulation both eyes at precisely the right location in those eyes, right? So there's an appropriate mapping of the visual field as imaged in the two eyes onto a common target cell. So I think that that degree of precision seemed to be greater than what could be achieved with genetic instructions alone. That was the thinking. Now, maybe they were underestimating the power of genetics, but the truth is that there are a lot of other mishaps that occur with the physical development of the body and the head and where the eyes are positioned. And so it kind of makes intuitive sense that there would be mechanisms in place to fine tune those connections based on visual experience, based now on postnatal visual experience to tune up those connections and
What you're saying is that there's one cell that's responsible, let's say, for some pixel in the visual field, and it needs to be tuned to exactly which pixel in the right eye and which pixel in the left eye are talking to it so that it can be stitched together.
that's exactly
So, like, if I
right.
obstruct one eye, I still see the same thing, in my
That's
final
right.
vision. Okay.
That's exactly
And
right.
there's small chance there would be no error in this. So you have to, as a baby, bring these images together somehow by adjusting the connection.
That's right. That's right. And those connections also, of course serve binocular vision in the sense that we see one world through two eyes, but also those connections serve stereopsis. That is the ability to detect depth.
Hmm.
Based. So it's critically important, the survival of species that require good stereoscopic depth perception, like a bat swooping down to catch a mouse. If they crash into well, bat's a good example. Come to think of
Okay.
it, let's go to an owl. An owl swooping down to catch a mouse, miscalculation, and they would crash into the ground. Right. So that was the thinking: was that this fine tuning process very likely involves visual experience, kind of learning. And the test of the hypothesis was a simple experiment as you can do in neuroscience, I think, which is that they close the eye in young animals. They could do it by suturing the eyelids closed. They could put a contact lens on one eye that blurs the image. In the early experiments, they could do with this manipulation we call monocular deprivation. If they did that for a week or 2 weeks or a month or so, and then they restored vision in that eye, what they discovered when they then went to cortex and recorded the activity is that the neurons responded only to stimulation of the other eye that we call in the field the fellow eye, the non-deprived eye. So it loses all the connections to the deprived eye.
And can these connections be restored?
They can be. And that's a really exciting, there's so many exciting questions that come out of this work, right? So the question is, well, how do you lose the connections? How do you restore the connections? And one thing I will say is that both losing the connection and restoring the connection is possible, but only during a finite period of early postnatal life. And that is what we call a critical period. Okay. There are critical periods of many facets of brain development. This is an example in the visual system, the best studied example.
And how long is this period lasts?
It depends. It depends. Yeah. In humans, in a way, it has to be operationally defined as the period of time when a certain manipulation can produce a certain effect. Okay. If that makes, I'm being a little cagey here. If you were to recreate in a human the conditions that Hubel and Weasel used in they were originally working in cats and also in monkeys, that critical period ends before the first year of life. So it's a very early critical period. If you do more subtle manipulations of visual experience, you can still get very robust effects, but then those can be extended out to 7 or 8 years of life. So it's so loosely defined. Typically, if you read about it, they'll the claim will be that the critical period ends at around age 7 in a human.
And what kind of processes define this critical period?
So let me be a little bit more explicit about what I'm talking about too. There are two ways to probe a critical period. I mean, these are the ways we do it in animal models. One is to define the age range at which commencing with monocular deprivation changes the responses in cortex. Does that make sense? So in other words, if I were to deprive an animal of vision in one eye for a week, and I started that at different ages, I could map out what age does that manipulation no longer have any effect. So that is one definition of the critical period. So that would be, to be really precise, it's the period of vulnerability to deprivation. The flip side is you've monocularly deprived and then you've reopened the eye and done a manipulation to promote recovery. How long is that possible? At what age is that no longer possible? Now, fortunately, both of these measures of so-called plasticity overlap to a great degree. But I think it is also important to be precise in how you're measuring the critical period because the rules need not be the same for vulnerability and recovery. So I think your deep question, and the deepest question of all, I suppose, is what brings a critical period to a close? Is that your question?
That's right. Exactly.
Well, this has been a question that has challenged a couple generations of neuroscientists. And I don't think there's going to be a single simple answer. I can tell you what's the prevailing theory, which is the prevailing theory is that inhibition in the brain is late to mature. And when it achieves a certain level of maturation, that ends the critical period. And this hypothesis is well grounded with data. I think what's less clear is what is it about inhibition that's constraining the type of plasticity that's present. So I think there's still some questions, next level of questions that are really important to address, but you can sort of break it down in a way. The way of thinking about it is there are patterns of activity that are arriving in the cortex. For instance, in the case of monocular deprivation, you have well-correlated patterns so neighboring ganglion cells are active at the same time when you're looking at the visual world, right? You're seeing shadows cross the retina, neighboring cells are active at the same time. We call that correlated activity, right? The other eye, an eye that's been deprived, that we would call that noise. It's not no activity. The retinas are quite active in the dark. Retinas are active unless we do something active to get rid of that activity. But it's patterned activity we replace with noise. So there's a process in the brain that is making a judgment about whether a synapse should be strengthened or weakened depending on whether it's a patterned input or a noisy input. At a basic level, that's the way it's got to work, right? So that is a very robust process during early postnatal life, and then it fades with age. So how come? So one possibility is that the molecular mechanisms of the synaptic plasticity that we'll undoubtedly talk about change with age. So, proteins that are expressed early in development are no longer expressed in adults, and that just the whole system sort of freezes out. That's one possibility. Another possibility is that these patterns of activity are no longer gaining access to the modifiable synapses. And that's how I view the role of inhibition, because inhibition can constrain the relay of activity in the cortex from one layer to the next and so on. And if that activity is not making it to the modifiable synapses, it's not going to have any impact on those modifiable synapses. Another hypothesis that has been around for a long, long time is modulatory inputs to the cortex, the inputs that convey information about salience and whether these modulatory systems are dialed down with increasing age. So a lot of very interesting hypotheses have been floated around for a long time. I think they're all going to be, in a sense, correct, right? So I don't think any has been excluded But probably the one that gets that's the textbook version right now is something to do with late maturation of inhibition. So let me say a word about that. So brain development occurs in waves, and very early in development, there's a creation of excitatory synapses. For instance, we talked about lateral geniculate nucleus and cortex. So those LGN neurons give rise to axons. They form excitatory synapses onto cortical neurons, and that's how we see. And there's a great deal of synaptic proliferation early in utero and after birth. It goes crazy just to get the system wired up.
How do people know this? Like, how do you actually study this? Do you have to freeze dry fetuses and slice them into little slices and look in a microscope? Or like, how do you know that this is happening?
All these studies have been done. Yeah. Anatomical studies, electron micrographic analysis, looking at synaptic density. One of the most amazing.
Is it something you can study on a live animal, or it has to be done by like actually
you
slicing?
can nowadays.
Okay.
It's a question of access and how long you can image. But nowadays it's amazing what can be done. So you can actually watch synapses as they're formed and follow them for a long time. In fact, I have colleagues right down the hall that are doing on a developmental time scale. That is, we're talking about months. That's a little harder. But fun fact is that there's a overproduction of excitatory synapses in the visual cortex. So they will peak at about 150% of the adult value in early childhood. And then there is a mad process where those synapses are whittled away during adolescence. So you lose about 50% of those synapses during adolescence as you're fine-tuning your connections.
So it's interesting. Fine-tuning or learning math in school is just about pruning.
Yeah, sure. Sounds good. Yeah, that's it. Problem solved. Okay.
Cut down the rest.
That's right. The more synapses you lose, the smarter you are. Quote me on that. So we've just been talking about excitatory synapses, but there is, in addition, everything you get in the brain is a balance of excitation and inhibition. And in fact, crudely, we always talk about what's called the EI ratio, the ratio of excitation to inhibition. So the EI ratio favors E early in development, but there is the late arrival of inhibitory neurons. The neurons themselves have a different migratory path to get to the cortex as it's developing, and the connections are slower to form. So the full maturation of inhibition coincides with roughly the time when that synaptic pruning stops. So these are conspicuous coincidences that inhibition matures at about the time you lose plasticity. At least in mice, it's possible to manipulate that development of inhibition, and you can manipulate the duration of the critical period in some fairly predictable ways. So that's what's really given the most support for this hypothesis.
So you mentioned that in your experiments, one eye is the fellow eye, like the working one, and the other one is the lazy one. So what happens for people who have both impaired eyes or maybe even like blind person, how the brain develops?
Well, I think one of the things that impresses me is that you think about, say, monocular deprivation as being a deleterious effect because you're losing responses to the deprived eye. Looked at it another way, the deprived eye is not really conveying any useful information. You're not forming crisp images on the retina. It's just a bunch of noise. And what you're really doing is taking maximal advantage of the neural hardware that you've got by decreasing the weight of that input and increasing the weight of the input from the fellow eye. So it's a compensation.
Mm-hmm.
And in fact, anatomically, this is very dramatic in the human visual system and the monkey visual system, where what you see is the amount of brain tissue that's devoted to the analysis of the eye that had been deprived shrinks quite dramatically. And you get a compensatory increase in the brain tissue that's devoted to the analysis of the other eye. So no brain is going to waste. And this is also true with congenital blindness. Like the regions of the visual system that normally serve vision are now being used to serve other sensory modalities. So the brain doesn't go to waste.
So something else takes the
yes,
the usage of
it is an area of neuroscience that I'm not following closely, but there are a lot of examples of enhanced, and to give an example, Braille, for instance, the ability for blind people to read Braille. I mean, that is a very fine tactile skill that is very difficult to achieve for a normally sighted person. And yet it can be done because you're really taking full advantage of the cortex that otherwise would be devoted to the analysis of the visual world.